Right on. Yeah, so I'm excited to be here and talk about some of the research that I'm conducting sort of in the region, particularly around urban ecology. And I hope to get across to show you some of the sort of research we're doing, but how that applies to sort of like everyday life and applies to wildlife management and conservation. And even maybe you'll see that we sort of work in the realm of trying to make cities a better place for people, even with through nature and wildlife. So for this presentation, when Luke said you can talk about whatever you want, I was trying to think of what I might talk about. And I sort of ultimately decided that since I'm new to the University of Maryland, and relatively new to the region, you'll see I've been here for a few years, but I am relatively new, that maybe today's talk will be a little bit more of like an introduction to myself and what my research team does. And in future Wildlife Wednesdays, I could come back and talk more in depth about particular topics that you all might be interested in hearing about and dive in deeper. So today's talk is going to be really like an introduction of where I came from, what I do, why I study urban ecology and like why I like urban ecology, and then give you all sort of just a snapshot of some of the work that we do in our research group and really focus around these three themes of urban ecology. And I'll talk about them in a minute. So it's going to be sort of a smattering or like highlights of research and showing how that research is applied to everyday life. So first, just give me a second to introduce myself. I started and I became sort of interested in wildlife or in sort of in nature in my early days as an undergraduate college student at the University of Texas A&M Corpus Christi, so a small branch of Texas A&M down on the Texas coast. As an undergrad, I went there to be pre-med and be a doctor and make a whole bunch of money in life. And then ultimately, through some different opportunities, decided I really wanted to work with animals, or at least like work outside. I had an opportunity working with the Marine Mammals Training Network down there when I was like in my late teens, early 20s, and got to rehab this rough-toothed dolphin, and then got to take this really incredible trip where the Air Force took us on a trip on a CJ-30 from South Texas to Florida to transport this dolphin to an aquarium. So there was some informative times there that decided to make me switch my focus from pre-med over to being, or at least trying to find jobs and working outdoors and working with animals. I ultimately stuck around that area in Corpus Christi and went to grad school there for my master's. My research was looking at Mexican ground squirrels and seeing how climate change might change their distribution. So it was like early species distribution modeling and predicting how their distributions might change as the climate changes over the next 50, 100, and 150 years. And while I was doing my master's, I took some side jobs that were really informative and really sort of sealed the deal that I wanted to be a wildlife ecologist when I grew up. I did some snowy plover surveys along the Texas coast, working for USGS. This picture in the bottom left looks like I'm sitting on a beach with white sands, but that's actually these salt lakes that are down in South Texas that are key to shorebird migration. And so that's actually like layers of salt there in sort of this inland lake in South Texas. Also worked with Audubon Society down in the Texas coast, building these waterbird roosts on spoil islands, trying to build some structure. So these sort of opportunities of really, and this sort of time in my life was informative and saying like, this is what I want to do when I grow up. I then couldn't find a wildlife job, but I got a job as my first professional gig in Austin, Texas at the Lady Bird Johnson Wildflower Center. Some of you may have heard of the Lady Bird Johnson Wildflower Center. They have one of the best online databases of native plants and where to get them. So if you're a native plant enthusiast, you can look at the Native Plant Information Network, or maybe you've used that. So this was part of the University of Texas, and there I actually ran a citizen science project called the Invaders of Texas, where we trained people all throughout the state to go out and look for invasive plants in their area and to map them. And we were really trying to figure out where these plants actually were, also training a big group of people to be early sort of eyes in the field for early detection of invasive species. And this really sort of was really informative in my life because I was working with people a lot. I went from being sort of in remote areas in South Texas to working with a lot of master naturalists, the Texas master naturalists, and volunteers, and training them, and really seeing the importance of sort of communicating science and conservation to to the folks that, enjoy it in their backyards, and maybe they're not also scientists. So this was I like to talk about this sort of first professional gig because it was pretty informative in my life. I then went on for more grad school at Colorado State. I was in the Fish and Wildlife and Conservation Biology department there. This is where I got my PhD. And my research project there was really focused on thinking about land use change and how that impacts wildlife. So really starting to get into that more conservation-oriented research. We specifically for my research, we think about these land use changes as things like urbanization, agriculture, energy development, deforestation, right? These are the common biggest sort of causes of the biodiversity decline and stuff. But out in Western Colorado, or in the West in general, it's a little bit here in the East, but really a lot out West, there's a big land use change that is focused simply on creating better habitat for specific species. So species you can hunt, like mule deer and elk out there, or species of conservation concern like the sage grouse. And we're talking, managing wide swaths of land, changing the landscape to promote habitat for these individual species. So a lot of my research for my PhD was using camera traps and counting birds in these areas where they were manipulating habitat for mule deer and sage grouse to see how the rest of the wildlife community responded to this sort of single species management out there in Western Colorado. And as you might imagine, results were mixed, but basically if you change a woodland, juniper woodland forest to a sagebrush grassland, over time, you no longer have woodland species, you have sagebrush grassland species, and so on. So we didn't see like huge declines in wildlife, but we did see community changes, which is just, goes from an applied perspective is important to think about when we're thinking about wildlife management and conservation as a whole. So this really got me thinking about what my future would look like and thinking about land use change and how it impacts wildlife. So sort of these like these large scale anthropogenic disturbances and how humans have an impact on wildlife populations. And I had this opportunity after grad school to do a postdoc. It's that sort of purgatory between finishing grad school and getting a faculty job where you need to go out and do your own research and get some papers published, thinking about urbanization and how urbanization impacts wildlife. And so I landed a postdoc at the Lincoln Park Zoo in Chicago and worked for this little institute within the zoo called the Urban Wildlife Institute. And so I was there for 3 years as a postdoc working on this big camera trap project in Chicago. So this is how our study was set up in Chicago. If you're familiar with Chicago, we have downtown here. This little airport that my mouse is hovering on is O'Hare. This other small airport down here is Midway. So we had these study sites across these 3 transects that went from downtown Chicago 30 miles out into the suburbs. And we had 110 sites. We had camera traps there, placed there, and we were really studying basically who lived where and why, and how do we take that information and apply it to urban planning and urban development so that we can restore green spaces to be more wildlife friendly and have overall more wildlife friendly cities. And so, did my postdoc there before I came here. And like I said, we used camera traps. So a lot of the data were photos like this. And some of these coyotes are from actually downtown Chicago. Some of you all might be familiar with the Chicago coyotes. But this is a big part of what my research is now and is really because of this sort of postdoc. So we had this study design in Chicago. And while I was there as a postdoc, we started to try to build out this across multiple cities. So we wanted to create this sort of camera trap study design that we could ship off to other cities. And we were forming what is called the Urban Wildlife Information Network. And so when I was a postdoc in Chicago, this is only about 5 years ago, we had 5 cities that were partners with us. They were doing the same study. They were putting cameras out on the sort of urban to suburban or rural gradient. They were putting their cameras out at the same time of year that we were, and we were sort of creating this like research collaborative where we were systematically doing the same studies across cities. Now to this day, we have 43 cities, and we are international. We have stuff in South Africa and Germany, in New Zealand, and even in Mexico now as our newest partner. And so when I was looking for jobs after my postdoc, a big thing that I was pushing was that I would bring this sort of same study design to a new city. And ultimately, I got a job at George Mason University back in 2019 and brought this sort of UN study design to this region, to Washington D.C. And then last fall, I moved over and transferred over here to University of Maryland and now faculty in the Environmental Science and Technology Department, but the research is still the same sort of study. And so this may look a little different than the Chicago study design. We sort of set a grid across the region, but in every one of these study sites, we put out a camera trap or a wildlife camera, right? A remotely triggered wildlife camera. You can see we have sites in the core of DC. We have sites in PG and Montgomery County, Maryland, and also down in Alexandria, Arlington, and Fairfax County. So we have 75 sites. They stay out for about a quarter of the year, and we've been doing this for 4 years. So it's designed sort of to be this long-term study. We have the infrastructure. As long as I have grad students and people that can go put the cameras out and collect the SD cards, we want to have this sort of long-term study. So the cameras go out every time at the exact same tree, the exact same site. And we're sort of trying to build this sort of grassroots long-term ecological research project here in DC. And starting off sort of with camera traps. And I have to give credit to a PhD student in my lab, Mary Collins, who joined me right when I joined at George Mason and really helped develop this project and get it going. And she actually moved over to the University of Maryland with me. So she's in my lab here at the University of Maryland. And she's been pretty integral in getting this project set up in the DC area. So some of the research I'll talk about today is coming from this project and some from some particular other ones, but this is really like my flagship study that I've got going on in the DC area. All right, so that's a little bit about me, starting from the early days to where we are now. But I want to step back just for a second and talk about why I like urban ecology, why I am fascinated by urban wildlife and things like that. And so, like I said, I am generally interested in how land use change impacts wildlife and urbanization. When we think about these four big players of land use change, urbanization is one of the biggest, right? More than 50% of the global population live in cities. 10% of the Earth's surface at the current time is covered in urban land cover, and it's the fastest growing form of land use change. So really, when we think about these big players, urbanization itself is the greatest human impact on the planet. So it's a big deal, right? There's no denying that. And undoubtedly, urbanization is changing the ecology and the nature of those areas where it is developed, right? But the really cool thing about urban areas is that they themselves are ecosystems, and ecological processes still exist in cities. We still have hydrology, we still have animals in cities, right? We still have soil properties and soil ecology and microbes. So ecology still exists in the city, except more and more research is coming out to show that some of these sort of ecological theories and principles that we use in wildlife management or wildlife conservation don't always hold up in cities. There's something about cities that, while ecology is happening, those sort of theories and principles we formed in more remote or rural areas aren't always generalizable to cities. I'll let you take a guess but I think it's probably people, right? These are human dominated systems. So I'm generally, from an academic or intellectual perspective, just fascinated by urban ecology for that reason. We can go out and we can test these sort of foundational principles in our field and see if they hold up in cities, and if they don't, why they don't hold up, and how we can use that to sort of inform wildlife management and conservation. And so just generally, I'm kind of fascinated in urban ecology from that aspect. It's like sort of a whole new novel world of ecology. Additionally, as a conservation biologist, the sheer number of people that live in cities offers this sort of untapped opportunity for conservation and conservation outreach, right? So if we want to really promote this sort of global conservation ethic, and more than 50% of the world live in cities, we probably need to get these urbanites jazzed about wildlife and nature and biodiversity around them, right? So any sort of research that I do in cities is directly connected to people. If I'm telling someone in a city a story about some findings that we found in our research, they're interested. It's not some far off place in Colorado or Canada or Alaska. It's right in their own backyard, and they can sort of picture that and be really in touch with with nature and the ecology around them. And so I think that I really enjoy working in cities because of the people and sort of the connection you can make with your research. Additionally, more research, I mean, for a while now, lots of literature and research has shown that urban green spaces in general, or nature in cities, is really good for humans. It promotes happiness. Green spaces offer places for recreation so it makes healthier people. It cleans up pollution and keeps areas cool. So it's really good for human well-being. And more studies are coming out showing that these same green spaces actually do provide habitat in cities for wildlife. And so there's no wonder that cities and modern urban development are really trying to incorporate more and more parks and green spaces into those designs. So I really hope that our lab really looks at the intersection of this and thinks about how we can incorporate ecology into that urban planning, so that we are making green spaces and we're making habitats more hospitable to wildlife, but also so that that can form a connection between humans and nature, even in these groups that aren't often thought of as like experiencing a lot of nature, right? So that is sort of the underlying motivation behind our research group, what we call the Urban Nature Lab. We don't actually pronounce the X in our logo. It's supposed to resemble or indicate the sort of interaction between what is urban and the nature that is in urban areas, that interaction between people and nature, and that they're not disconnected. So we're the Urban Nature Lab. Our vision is cities where people in nature thrive. And that is sort of the motivation that motivates myself as the lab PI, and also I think the students and postdocs and researchers that work in my research group. So I just want to, from here on out, sort of give you all, like I said, a highlight of some of the research projects that we do in our lab. And I've got a lot of students right now, maybe too many, running myself ragged, but I'm really highlighting today some projects that are local in the area and that are pretty much wrapped up and finished up, and ones that I find really fascinating. So I will frame these projects in this sort of overarching paradigm that our lab sort of follows. And this is from Stuart Pickett's. He's up in Baltimore. He's actually at the Cary Institute, which may not be directly in Baltimore. But he's sort of one of the founders of this field of urban ecology, and he created this urban ecology paradigm. And it's the idea that we have sort of three approaches to urban ecology. You have urban ecology in the city. So this is the idea that green spaces are analogs to nature. So we're sort of doing similar ecological research that we would in a national park or in more rural areas. We're just applying that line of thinking and research to cities. So we're saying, what is the ecology in cities? There's the ecology of cities. And so this treats the city as an entire ecosystem. So incorporates both the man-made and the biological aspects of the city and thinking about how they're all interconnected. And then ecology for the city. So incorporating human well-being. So how are we taking ecological processes and applying them in cities so that it makes better lives for urban residents? So I'll try to frame a few of these research projects under these three themes that our lab follows. So first, thinking about ecology in the city, I'm only going to highlight one study that we just sort of wrapped up, and it's sort of out there and getting peer reviewed by other scientists. But this is looking at turkey ecology in the city, and specifically DC urban turkeys. So turkeys were almost extinct in the eastern US. They were extirpated from Virginia around 1920 and almost extirpated from Maryland. But since about the 80s, there's been major conservation efforts for turkeys in Maryland. And it's been a really big success story. It doesn't get a lot of attention, but I think the turkeys have been as big a success story as bald eagles and brown pelicans and things like that. And we're actually starting to see those successes and those reintroductions that Maryland have done spill into the urban areas. And wild turkeys started becoming sort of redocumented in the urban areas of the DMV around 2005 and 2006. But to date, really, there isn't a lot of research out there on urban turkeys and turkey ecology in cities, these urban turkeys. And this is a project that is led by Mary Collins, who I already sort of introduced. And turkeys have been making the news the last few years. If you've been following, they've been sort of wreaking havoc in Boston and Pittsburgh on some of the college campuses. And they've made the news on The Guardian, and the New York Times even had a story about these urban turkeys coming in and and messing with people. We even had our own story last year, right? This Tom, maybe you guys saw this Tom out on the Anacostia River near Kenilworth Gardens, was running out and attacking cyclists and walkers and stuff in that area. There was a Popville article about it where someone they drew blood and they had to get a tetanus shot and go get antibiotics because this Tom chased them around. And so this was going on in the news. We were talking to people at the National Park Service and other land managers in the area about turkeys, and just it kind of came up like, yeah, we don't really know a lot about urban turkeys and what their habitat preferences are, why they're in the city, where they are and things like that. We just know they're sort of around now and they're becoming more numerous. And, we have these cameras all over DC and the sort of the general area of DC, and we get a lot of turkey pictures on our cameras. These cameras, typically people study mammals with these cameras, but turkeys rarely fly and they run around on the ground and they're big. And so they kind of act more like terrestrial mammals than actual birds. And we get a lot of pictures. So we thought, hey, let's use our data and see what these habitat preferences are for wild turkeys in the region. And these are the sites that we see turkeys and some that we see turkeys more often. And so we wanted to see what is the probability that a turkey is present at one of these sites given some environmental characteristics. So we looked at things like tree cover, shrub cover, tree height, distance to road, distance to water, human population density, the patchiness of the environment. And we wanted to look at those sort of parameters and see which influence the probability that we would see a turkey at any one of these sites. And so what we found actually is that the two variables that had the strongest and significant correlation with turkey presence was elevation and distance to road. So there was a negative correlation between turkey presence and elevation, and a positive correlation with distance to road. And so habitat at lower elevations in the city and further away from roads had higher probabilities of a turkey being present at that site. And so this is kind of this was interesting to us because where is the Anacostia River Trail? It is at lower elevations along the river and pretty far away from roads relative to some of the other sites where we have cameras. So that made total sense. This is where we have turkeys. So this can be used, right, by local land managers. Turkeys are still a species of conservation concern in a way. So people think about turkey habitat. So if we want to sort of restore or create green spaces that promote turkey habitat, we might focus on those areas that are closer to lower elevations, which generally are floodplains, so closer to the rivers, and also areas a little bit more isolated from roads. But at the same time, from like a human wildlife coexistence perspective, if you have a hike and bike trail or some sort of area of recreation going through those low elevation areas, you might think that this would be a good place to put up some signage, right? Or some educational literature that says, hey, this is turkey habitat. This is what you do if you see a turkey, or if a turkey comes out and seems aggressive, right? So you could target some of this educational outreach to these areas where humans are recreating in the area, and it's also good turkey habitat. So this is one of the first studies really to look closely at habitat preferences of urban turkeys. So this is an example of ecology in the city, right? What are turkeys doing in the city? If we want to look at a couple examples of ecology of the city, I've got two projects here I want to highlight, and the focal species of this is actually domestic cats. So this may still seem like ecology in the city, and it kind of is. They're not mutually exclusive, but I think cats in the city are a human construct, and they are there because of humans. They are where they are because of humans, and therefore they kind of represent this sort of man-made disturbance and sort of that interaction between the human aspect of a city and the natural native wildlife of the city. So this is a project. It's actually two different papers wrapped up in one big project that was led by a PhD student in my lab who's here at University of Maryland, Dan Herrera. And so Dan wanted to look and see who were cats actually hunting. There's a big debate on if cats actually eat rats in cities and how much do they eat compared to native wildlife. I think everybody on this call probably recognizes that cats are have a big impact on native biodiversity. They hunt in native biodiversity, but we want to look a little bit closer at this rats versus native and who they hunt. So Dan used data from camera traps all over the city, and he took all the photos of cats and all the photo of cats carrying around a prey item. And he identified those prey items in their mouth down to the species level as best as he could. But surprisingly, there's a lot of cats in DC, and we have a lot of photos of cats, and we have a lot of photos with cats with animals in their mouth. So we actually had quite a pretty rich data set, even paring it down to this very specific of a photo, a cat with an animal in its mouth. And so we wanted to look at this idea, like, is that animal a native species or is it a rat? So that became the sort of data point we were looking at. And we wanted to see if that was related to some environmental characteristics, like distance to forage edge, impervious surface, or supplemental food that was left out. They had done a survey for this project. I'm not going to get into those results, but they had done a survey to try to figure out where supplemental food was or predict where supplemental food was across the landscape. So we're going to look at today is the results of this idea, is it native or is it not? So this sort of binary response, is it a rat or is it not? And seeing if that relates to distance to forest edge and impervious surface. Impervious surface being like buildings and roads. It's like an indicator of high density urbanization. And so what we found when we first look at that probability of the prey item being a native species is that the distance to forest edge was the strongest predictor. And it had a negative relationship to it. So if you look on this graph over here on the left, where you're really close to a forest, you have a higher probability of that prey species being a native species. Whereas if you get further and further away from the forest edge, you don't see that as much, and you don't see it being, you see a lower probability of it being a native species. Where around 500 meters from the edge of a forest patch in the city, you're kind of getting down into 0% probability it's native. So this was interesting and a very straightforward sort of result, right? Distance to forest edge is driving whether they're preying on native species or not. And then if we look at the rat results, we actually found the same thing, but opposite. So distance to forest edge was the strongest predictor of the probability that that prey species was a rat, except it had a positive effect. So it was a different model if you're paying attention to those models down here, but forest was still the strongest predictor compared to impervious and food supplemental food. So if you're really close to the forest, over here on the left, you have a lower probability of that prey item being a rat than as you get further and further away from the forest. And you get almost up to 100% probability that it's a rat and not a native species. And so this was, again, another great result, another straightforward result, because we can combine these to look at this from a more applied or practical perspective, because it's the exact same parameter that's driving this probability of it being native or being a rat. And when we lay them on top of each other, we see that right around like 300 meters, there is a switch where all of a sudden you have a higher probability of it being a rat than it being a native species. And so we can now have this sort of hard evidence that says, for at least for DC, that says, hey, maybe we should do some sort of restrictions or regulations right around that area where the probability switches, right? Or maybe, and so that's this map on the right. This is like exclusion zones or buffer zones that are around 300 meters from a forest patch in Washington, D.C. And so that's where we say, like, okay, the probability of switching from a native species to a rat. And the impetus for this was that, like, I think cats should be indoors, but that's not happening in any cities in the US. We've made no progress really in that sort of debate. And so we're trying to find some way to say, like, okay, well, if people aren't going to keep their cats indoors and we're not going to make regulations, let's do something that is at least lowering that impact to native wildlife. And this was sort of this idea of keeping these exclusion zones. And so this was the switch, but you can sort of take this information across any of these predictive values and say, okay, we'll come up with our own value judgments as policymakers or managements for this. So if we want it to be 1% probability of a native wildlife or less, we can come back over here to this graph and see that, we need to be about 500 meters from the forest. And so we can make a map of a 500-meter exclusion zone. Maybe you're okay with it being 25% probability, which is still, a lot lower than chance, right? Then we can have smaller exclusion areas, or maybe chance is like the value that managers are okay with. Like if it's less than chance, 50% or less, we have these exclusion zones. So we're probably not going to go in there. Like I would imagine that a city is not going to say you can't have cats outdoor in these areas, just from historic, policy of feral cats. But we could easily have policies that say things like, we're not allowed to have sanctioned cat colonies within these buffer zones. We're not allowed to do trap, neuter, release in these buffer zones. If you catch a cat in these, they have to be removed from those buffer zones and euthanized. And you can say, okay, well, elsewhere in the city, we'll have looser regulations for residents because they're having less of an impact on native wildlife in those areas. And so we can have these very sort of straightforward recommendations for feral cat policies. And this would be an example of including a biological aspect to it too. So saying, here's a 25% buffer zone, also adding the home range of a cat. So it's always excluded from the buffer zone, right? And so here, if a manager or a policymaker is okay with it being 25% or less probability of native species, you could have some buffer zones that look like this across Washington, D.C. And these these green patches are what are considered forest patches from the urban forest index. So this would be some sort of regulatory map. And this paper is published in Biological Conservation. You can go read it if you want to get some more in-depth info on the methods or the results that we found for this particular study. So then we were still thinking about, or Dan was still thinking about this sort of idea of cats and this idea that we're not going to exclude them. So how can we have more fodder for this idea of keeping cats out of native habitats? And so we had this second research question thinking about when and where do cats and native wildlife overlap? And so think about this from two perspectives: spatial, so where on the landscape, and also temporal, so where in their daily activities do they share active time? And so we can think about this from an individual species perspective. And just like that turkey project, think about what's the probability that a native species is in this part of the landscape given some environmental characteristics. And what is the probability that a cat will be in one of these areas of the landscape based off its own set of environmental characteristics, because species select habitat at species specific levels. So we can sort of understand the habitat preferences of the two species and then see where those spaces might overlap and calculate a probability that they would be located in the same location based off of basically just multiplying the probability that they each individually are in a particular location. So that could give us an idea of this probability of shared habitat across the landscape. But also we want to know about, maybe they're at the same spot, but maybe they're active at totally different times of the day, and so they're not really coming in contact. So we also can go at the temporal overlap using our camera traps, because our camera traps come with the time that photo is taken. So we can see when we get the most photos is an indication of the time of day that these animals are more active on the landscape. So we can look at individual daily activities for the native species and the cats and then see when those times might overlap. So to see those shared active hours. And the context for this research was really thinking again about this sort of compromise across the landscape. So we know that cats can give diseases to wildlife. They can directly hunt wildlife and kill them. And they can create this sort of fear response that makes wildlife maybe not go to in optimal habitat or changes their daily activities. So the sort of like non-mortality effects on wildlife. But at the same time, and this isn't often in the discussion among us as conservation or wildlife ecologists or wildlife enthusiasts, cats can be harmed by wildlife. Wildlife can give cats disease. Certain wildlife can harm cats by attacking them and injuring them or even killing them. And they also can be hit by cars or, eat poisonous things in the environment and things like that. So there is, when wildlife and cats overlap or come into contact with each other, there's an inherent dangers for both of them, right? So obviously leaving your cats indoors and them not interacting with wildlife eliminates all that, but that's not happening in cities. So we wanted to come up with this sort of suggestions based off of this shared space and shared time to show that, these animals shouldn't be overlapping with each other. And so that's sort of the motivation to this particular project. So we focused on a suite of species, 3 species that can cause harm to cats: raccoons, foxes, and opossums. We called them vectors because the opossums aren't really attacking cats, but they can share diseases back to cats. And then we had this suite of prey species, white-footed mouse, chipmunk, woodchuck, eastern gray squirrel, and then cottontails. So we did these individual cat and individual species temporal and spatial overlaps for all these different sort of combinations of species. I'm only going to show examples of 3 of the results from this particular research project. So first, here is the results of the domestic cats. So I'll walk you through this a little bit. These are the probability of a cat being on the landscape based off of some environmental features that were driving that. And we did this at 3 scales. We have the finest scale on the left, and then we have it all the way up to 1 kilometers sort of scale on the right. The darker, the cooler colors are lower probability of a cat being in that present in that sort of cell based off of environmental characteristics. And the warm colors, so the yellow, are higher probabilities. And so you can see from the map on the left, high probability of cats being on the landscape in like that urban core of DC, less so as you get into the deeper parts of like Rock Creek Park and some of these bigger forest patches. And that's because cats were really driven. They had a negative relationship with water and canopy cover, which is the W&C, and a positive relationship with human population density. And so we're going to see some more of these maps, but warm colors mean a higher probability of that species being in that cell, that little cell, and blue colors, cooler colors mean lower probability. And then we have this activity pattern on the right. And so with this activity pattern, we're not going to get too in depth with it. You'll see how they overlap with other animals. But what this particular thing shows with cats is that as a collective species, cats are active 24 hours a day in DC. That's not an individual cat is always active 24 hours a day, but as a species in DC, cats are active 24 hours a day. You have night cats, you have day cats, and there's always out there. So already with this one result, you're going to see that cats are always have a temporal overlap with native wildlife, and so both vectors and prey species. And so if we combine that with individual species, we'll first start with the red fox. And so we have red fox specific maps on the top. And so you see that in sort of the deeper areas of the forest patches like Rock Creek Park and Kenilworth and things like that, we have a higher probability of foxes being present, not so much in the urban core, but these aren't completely zero. So we still have a probability of foxes being in these sort of landscapes. And so we can take these maps and combine them. We're basically just multiplying that probability together to get a probability of spatial overlap. And we see that in these sort of outer edges of the city that have more tree cover and more sort of forest patches, that cats and foxes are overlapping more in those areas spatially. There's a higher probability that they are in the same location. And then we see that there is this overlap. So the light blue shaded circle is the wildlife species, and the clear circle is the cats, and the dark blue is that shaded area. And this value actually under each one of these will be the proportion of that native species daily activity that is overlapped with cats. So essentially, cats overlap foxes' daily activity patterns by 73% of it. So that's a pretty significant amount of the fox's daily activity patterns that cats are also active during that time. So we see these areas of high spatial overlap within the city, and we always see that they are sort of temporally overlapping. So it's important to think about these locations and where cats are interacting with foxes, because foxes can kill cats. And then if we look at raccoons, as you might have thought, raccoons are found everywhere. Maybe the urban core is not as high a probability, but still there's probability that the raccoons are occurring in those areas. And so when we overlap that with cats, we see a pretty significant spatial overlap or higher spatial overlap of raccoons and cats across the city. And they overlap in time, 50% of their time is overlap. So again, you have this argument when thinking about cat management and talking to sort of cat enthusiasts, that it's like, well, your cats, when they're outdoors, they are overlapping both in space and time with foxes and raccoons. So your cat is in danger. And that was sort of the intent of this project, or the motivation of this project, is to show that both cats have inherent risk of being outdoors, so does wildlife. And we can show with data that, yeah, your cats are overlapping and sharing space and time with animals that can harm them. So they are also in danger when they are outside. We did this for a bunch of prey species, as I showed. This is just an example of eastern cottontails. We see that in sort of the northwest part of DC is where cats and chipmunks, sorry, I said cottontails, chipmunks. You can see this is where they overlap the most, and they still overlap almost half the time of their daily activity. So as we've all known, cats prey on native wildlife. They're overlapping with them as well. And so take home message from this, is cats are safer indoors, right? We know they attack wildlife. We saw that with the predation paper. We know that when they're near forested areas, they're preying more on native wildlife than rats. And now with this project, we showed that also around some of those more forested areas, cats are overlapping with their prey species so they can kill them, but they're also overlapping with species that can cause harm to them. And all of that risk goes away if the cat is stayed indoors. And so this paper is also published in Frontiers in Ecology and Evolution. So you can check out that paper as well. And then the final project I want to talk about is one that I think represents sort of this idea of ecology for the city. So what are we doing the work for? When we're understanding ecology, how are we applying that to make human lives better? And so I'm just going to highlight this one project we wrapped up looking at urban wildlife as sentinels, and more specifically looking at urban rats as a sentinel for lead exposure. And yes, I consider urban rats part of the wildlife community because there's so many and they are sort of ingrained in urban areas. And this research is headed up by a PhD student at George Mason that's in my lab, Krista Shires, and she's actually graduating tomorrow, so that's exciting. So she just wrapped up this project for her dissertation, and now we're working on getting it published. So the motivation behind this project is that a few research papers came out with the Flint, Michigan water crisis that showed that domestic dogs were great sentinels for lead exposures in their human companions. And this makes sense, right? Dogs are with you all the time. They're drinking the same water you're drinking. They're in the same environment in your house. They are sort of like the closest companion animal besides cats around that. And it showed that when a dog had high lead level, the person had high lead levels. And so they were saying that these domestic pets, could be used as sentinels for lead testing. And the idea is that it's logistically hard to draw blood from people. So could you do something on an animal that has less regulatory reasons to do this sort of testing? So Chris and I were thinking about this and thought this was fascinating and thought, well, what is another animal in cities that live really close to people? And it may sound gross, but it is true that rats are that next animal, right? Rats are everywhere in cities. They live in people's backyards. They live under houses. In more infested areas, they're living in houses. In the wintertime, we've learned that rats rely 100% on water sources in the walls of buildings and houses. And so they, in a way, are sort of drinking the same water. So besides dogs, rats are sort of the next species that live in really close proximity to a lot of urban residents, and nobody cares about rats. We catch them, we exterminate them. Therefore, they are a species that we could probably use as sentinels because we could catch a bunch, test their blood for lead. And if they have high concentrations of lead, maybe the humans nearby also have high concentrations of lead. So that's the motivation for this project, was to see if to find some evidence that rats might be sentinels for lead exposure in humans. So Krista worked with a company out in Virginia that collects rats in DC using dogs. So the city of DC doesn't actually trap and kill rats. They just throw out bait and poison rats. This company actually uses these little dogs here to chase them down and collect them. The dogs kill them right away and they collect rats. And so they have a lot of commercial and residential customers in DC, and they were super jazzed to work with us because they found this idea fascinating. And in a way, this family business are rat enthusiasts. They are crazy about rats. They have a strong respect for rats, which I actually do now after this project. And so they were keen and jazzed to work with us on this. So they had all of last year, they saved their rats that they caught for their customers for us, and they marked the cross street of where they got those rats. They weren't going to give us their customer's address, but they marked the cross streets. So now we have rat specimens and we have the geographical location of where they came from. And so these folks collected them and saved them for us in a freezer in their Woodbridge, Virginia facility. Krista would go get them from them, dissect them up on George Mason campus, get their livers, send their livers off to a veterinary lab at Penn State, where they would give us the lead concentrations of these individual rats. And so that was sort of the methods for this. And so here is just some summary stats of the rats that we collected throughout DC. These are the locations where we got rats, calculated the mean lead level of these rats that were collected within the different 8 wards of DC. And if you look at the CDC lead criteria, you see that if it's above 0.35 to 0.59 parts per million, that is indicated of lead poisoning. So that's like toxicity level. And you see that all 8 wards, except for Ward 3, which is like the Georgetown sort of southwest ward, had rats where the average lead level was above lead poisoning in these rats. So that already was striking to us. Then we wanted to take these lead levels in rats, try to correlate them to some environmental variables that related to sort of humans. So road density, distance to water, the human age in the neighborhood, the proportion of non-white residents, and seeing if there was actually a racial divide in lead content in the environment or in the rats, building age, income, distance, landfill, and kids under 5. So I'm running a little bit out of time, but I've only got about 3 more slides. So I'm going to wrap this one up. And then we just wanted to look at the correlation between these environmental variables and the lead levels in rats. And what we found was the only 2 significant predictors of lead levels in rats was distance to landfill and building age. And they both had positive relationships with lead levels, and building age was actually the strongest predictor. So as you got further away from a landfill, actually I have some graphs here. So as you get further away from a landfill, you have a higher lead levels in the rats. So this was surprising to us because we thought rats near landfills would have higher lead levels because landfills, and so we use landfills, incineraries, any sort of those dumping grounds that we could find in the DC area. We have a hypothesis that we need to dig in more, but we think maybe there's so much regulations around landfills that those rats near landfills actually are benefiting from those regulations. But as you get further away, there's less regulation, so more lead in the environment. I don't know. This is still was surprising to us, and we're still trying to work out a hypothesis for this. But building age was the strongest predictor, and a pretty strong predictor. And you see that as the building ages in the neighborhoods where these rats came from increase, those rats had higher lead levels. And these dotted lines are our 95% credible intervals. So 95% confidence that our parameter would land in the red in rats having lead toxicity when buildings are around 90 years old, which is pretty much the common building stock of DC or older. And so this was really strong evidence to us, that more evidence that rats could be used as sentinels for humans. Building age is directly related to housing. Housing is directly related to humans and the environment that they live in. And we see higher lead levels in rats in near older buildings. So another sort of step of that evidence of like, hey, maybe we can use rats as sentinels for lead toxicity in humans. So the next step for this project is to take those rat samples that we have and connect them directly to human lead level data. And there is data out there. The city has lead levels of children ages 2 and 5. All those children in DC have to get tested for lead. The city has that data. And we want to sort of correlate that both spatially, but directly across the landscape to see where these areas might have stronger relationships. And so this was the original intent of this project, but we actually worked with the city for a couple of years talking to them about that to get this de-identified data. And then ultimately at the end, they just said, no, you can't have it. And so we're still sort of working on figuring out how to get that data. So we shifted to thinking about environmental characteristics that related to humans instead of being able to directly connect this to human lead levels. But this is sort of the next step for this project to really show that rats might be able to be used as a sentinel. So that's sort of a snapshot of what I do. I can dig into the deeper topics at a future Wildlife Wednesday. I'd be happy to. But I just wanted to sort of introduce myself as a newer research in the University of Maryland community and show how our lab's work really focuses in that intersection of ecology and people and policy and planning and urban development, which I think is the tenets and the principles of urban ecology itself. And so with that, I want to say thanks for coming to this talk. Here's my contact information. Feel free to email me anytime, and I'll take any questions.
Thank you so much, Travis. I really appreciate you sharing. I learned a lot about the breadth of your work. It's really great to hear it from firsthand. So everybody in the audience, we had a couple come in on the chat, but feel free as you're listening here to pop any questions in the chat that come up. Jay Everhart asked the same question that I was thinking about, is what proportion of cat prey were birds? And I was also thinking about, well, and also what other types of wildlife, the spectrum of native versus non-native, and native mice versus, I guess there's some non-native mice and things like that. So I'd love to hear some of that data.
Yeah, let me actually stop sharing because conveniently enough, I have those numbers. And since we're doing Zoom and technology, I have those numbers right in front of me from Dan's study. I just got to pull it up real quick.
While we're waiting, everybody, I will pop a link to our YouTube channel where this will come in. Again, feel free to click that link. And if you want to subscribe or like what you see, give us a like on there. It helps to spread the word about education efforts we're doing.
Let me see if he has that.
And
Yes, he didn't actually break it out into birds. He did in this presentation that he shared slides with me. He broke it out into just native versus non-native, but cats accounted for 77% of all predation events. So he's been doing this for all species. This was a cat study. So even compared to foxes and coyotes that we get with animals in their mouth, the cats accounted for 77% of all predation events that we have across all our cameras. And 12% of those events were on native species. So it was a larger proportion of rats than native species overall, but I think that's because we have a lot of sites in these like urban cores. But still 10% of our, at least where these cameras were native species.
All right. Cool.
Yeah, as I can give you a little summary stats. So of the species they found, the most common species from our cameras was actually domestic cats. This is detections. This isn't individuals. We're just looking at detections. So cats are really, really, really common in the DC landscape. So I found that pretty
more common than squirrels.
Yeah, more common, like right around the same. But we get a ton of squirrel pictures too. So that I found that quite interesting. And I mean, when we do night surveys, I've gone out with the city to do night deer surveys and stuff with them. It's pretty striking how many cats we see.
Oh, cool. Feel free to anybody else in the chat to type in their questions. We had, somebody, David Montgomery asked, can you suggest a coyote infested area where he can drop off his neighbor's cat?
Yeah, that's a funny question. But what an interesting segue from that though is just generally from our work that I find fascinating about coyotes is we don't get a lot of coyotes on our cameras in northern DC and into Maryland. We get some coyotes around at a site in the Maryland side of Rock Creek Park and on the Sligo Creek golf course. But like we've only gotten one picture of a coyote on any of our PG County sites, but we get a ton of coyotes in our Northern Virginia sites. And, coyotes are sort of advancing up the eastern seaboard from the south, and they are getting them up in New York, which they may be coming around from the Midwest there. And our partners in Delaware have only gotten one picture ever in 5 years of a coyote in Wilmington. And so I kind of think our region is on the forefront of this recolonization of coyotes in the area. And so we're seeing them all sort of in the south of DC, but not so much in the north, really low densities at least. We get them on camera, but the rate of getting them on photos is not super common. So it's an interesting question. I think I didn't talk much about coyote stuff in this presentation, but I find it fascinating. I've been trying to find funding to do a study of collaring coyotes and foxes and seeing how they interact in the city, because they already exist together in Chicago, and there's no foxes in Chicago, and people think the coyotes drove out the foxes. But in Madison, a much smaller city, they sort of coexist together. And DC might be one of those cities where if we're in early, we're going to see this recolonization of coyotes. So I'm really fascinated about coyotes. So it's a funny question, Sligo Creek golf course, but then I don't want you to go drop cats off at the golf course because there's birds out there, but it does sort of segue into my fascination about the urban coyotes in the area.
Yeah. Well, he mentioned, and this speaks to the importance of, or the impacts they can have. He said they've lost 2 bird nests, 2 rabbits, and a turkey nest to cats so far this spring. So, and he lives, I believe, in a pretty rural area on the Delmarva in Virginia, actually, I think. So,
yeah, I could I'd have to find it, but I could share it with Luke. My PhD student, Dan, who did these studies, is really got into this cat stuff, and he has on his sort of website these postcards he's made. He's also into graphic design and infographics, and he's made these anonymous postcards that you can buy from him on his Etsy shop and drop them in your neighbor's mailboxes or put them on their doorstep. So I can share that link with Luke if you are a passive aggressive neighbor, which I am, and I've done before. So
Oh, that is funny. That is fun. I will definitely like to see that. Cool. We have just a comment from John Hooven, our forester. We have eastern coyotes in the New Jersey pine barrens. So just a comment on where they're detecting coyotes.
And our colleagues in New York get them even in Central Park, and then off to the east of New York. So I imagine that I kind of think those coyotes have come over from like the Midwest coyotes, able to come over, and I think the ones we're getting are coming up from the Carolinas and stuff, and they're just kind of converging into the Mid-Atlantic maybe, but they're not here in high densities yet.
Cool. I know I had another question on this rats piece. That was pretty remarkable levels that they have in their systems. Well, I was thinking about it. Well, they are like running along corners and edges where dust and old paint chips and stuff accumulates, I guess, and possibly also in old water pipes. Gosh, I was thinking about, have you started seeing any of this stuff like move up the food chain, like with predators of rats? Like, could these lead levels start to accumulate moving up? And have you thought about that at all?
No, I haven't thought about that's an interesting thought. I haven't thought about it from a trophic perspective. I've been thinking about it more in this like sentinel ways, but I would imagine start testing cats for lead.
Oh, wow. Yeah.
Cats, right? Like,
Yeah.
because they're probably, I would imagine in the city, they're the main predator of rats.
Right. Huh.
Yeah, I want to explore. We need to dig a little bit more deeper into this data, and we keep getting pushback about sentinels because of what you just said, that rats are definitely like in the soil more than humans are, obviously at a higher concentration, and that's where a lot of lead is in cities. And they're like in the walls, so near lead paint and stuff like that. But I do think that, we might have that like type 2 error where if we don't see a positive relationship or like a positive correlation with humans directly, that may not mean that humans aren't have lead issues as well. But if we did see a strong correlation between the two, then I think it shows that they could be using a sentinel. And I could see that being a tool in urban areas where you could go out and do like a rapid assessment where you go to a building or a neighborhood and you catch a bunch of rats and you test them. You don't need a lot of permits for that, right? And then if you're seeing high lead levels in rats, you're like, okay, well, maybe need to like do some intervention here with the humans instead of just this sort of this like not proactive, the opposite of proactive,
responsive reactive.
Yeah, responsive reaction, whereas kids start showing up with lead poisoning and then we start looking at neighborhoods. You could do like a little bit of a rapid assessment type stuff. That's the idea next.
Have people aware and start changing behaviors and cleaning up their corners and keeping things tidier, I guess. But yeah, it's scary to think my daughter grew up and we were living in an older row house in the Alexandria area, and I'm sure there was old lead paint, from way back that was.
Oh, yeah.
But well, great. Well, we're a few minutes past 1 o'clock. Anyone have any final questions? Is your last chance, last call? With that, I want to say thanks, Travis. Really cool learning about what you guys are working on, and look forward to meeting some of your students in these labs in your lab one of these days. It'd be fun to visit and just do some brainstorming and socialization. So,
yeah, for sure. Especially now we're all kind of coming back in person over the last year. So excited about that.
Well, great.
Yeah, well, thank you for having me. I really appreciate it.
Yeah, thanks for sharing. And again, everybody, we're getting some thank yous coming in the chat. Thanks for joining us. We look forward to having you next time learning about oaks. John Hooven here will be giving us a session on oak trees. So I look forward to learning a lot from that as well. So thank you, everyone, very much. And we will see you soon. Take care. Bye-bye.